Welcome back. In today's class, we are going to learn about the structure of the bacteriophage and the life cycle of it. To speak about the structure, the bacteriophage is divided as the head, body, and tail, where there are many parts that are being included, which has its own role to play. The first one is the capsid head, which gives the structure of the bacteriophages and also encloses the nucleic acid of the bacteriophage inside it. This nucleic acid encodes for all the signaling processes and all pathogenicity. The collar, whisker, and the sheath is the intermediate between the head and the tail, helps in the adheration. There is something called as the base plate, tail fiber, and the spikes that are the part of the tail of the bacteriophage, which also helps in adheration, and also the spike creates a pore in the cell wall of the bacteria where they adhere to, so that the nucleic acid can be injected from the head through the sheath and enters inside the host. Bacteriophages are the viruses which infects bacteria. That is why they are called as bacteriophages. These are the ones which are the pathogen for the bacteria. There are certain beneficial role that bacteriophages play, such as sewage treatment, where certain pathogenic bacteria are killed using friendly bacteriophages. To speak about the life cycle, the bacteriophages show two types of life cycle, that is lytic cycle and lysogenic cycle. As the name signifies, lytic means there is lysis. As I told you, the bacteriophage adheres to the bacteria and causes death of the bacteria. So the bacteriophage attaches or adheres to the bacteria, tries to inject the nucleic acid from the bacteriophage inside the bacteria, enables it to replicate it rapidly, and there is assembly of this pro units, and then there is lysis of this bacterial cell and a lot of progeny of the virus that's been liberated, so that goes and attacks the fresh healthy bacteria. This is the overview of the lytic cycle. To speak a little bit in detail, when the bacteriophage attaches to the bacteria, that stage is called as the first stage, called as attachment or adheration. Here, the bacteria phage attaches to bacteria. Later, the nucleic acid of the bacteriophage enters inside the bacteria. That stage is called as entry. The next stage, the third stage, is called as the DNA replication and protein synthesis, where this phage DNA, that is the bacteriophage DNA, enters inside the host cell, that is a bacteria cell, and starts developing, starts replicating. Now, there are a lot of viral subunits that are being produced inside the host cell. Now, these viral subunits need to be assembled in the form of bacteriophage. That stage is called as assembly stage, which is the fourth stage. Newly assembled phages. This assembly, once this assembly happens and maturation happens inside the bacterial cell, they lyse and all the progeny of the virus has been liberated, which goes and attacks the healthy bacterial cell. This is called as a lytic cycle, where there is, as the name signifies, it shows lysis in the bacteria. So this explains how there is a penetration, as I know, attachment, penetration, protein synthesis, and there is assembly and the liberation of these newly synthesized viral particles. To study this lytic cycle, T4 bacteriophage is the best example, where the T4 bacteriophage looks like this, where it is divided as head, body, and tail. To this, once the bacteriophage attaches to the bacteria, the spike creates a pore in the bacteria so that this component of the bacteriophage shrinks or compresses each other so that the nucleic acid can travel easily through that and reach the bacteria. So this is the life cycle of the T4 phage, where there is adheration, there is penetration, there is protein synthesis, there is newly synthesized viral progeny, assembly of them, and liberation of the progeny. The next type of cycle that we are going to learn is lysogenic cycle, which is the second form of cycle where there is a small difference between lytic and lysogenic. In lytic, as the name signifies, when the viral progeny is being synthesized, the bacteria lyses and the viral progeny are liberated into the environment. But in lysogenic pathway, there is no lysis for the host cell. When the bacteriophage enters a prokaryotic cell, that is a bacteria, the nucleic acid is liberated inside the bacteria. That prophage enters or integrates inside the nucleic acid of the bacteria. As we know, the way the bacteriophage has nucleic acid, even the bacteria has. So here what happens in lysogenic pathway is that there is integration of the phage DNA inside the host DNA. That is, the bacteriophage DNA integrates inside the bacterial DNA. Then as the host divides, even the prophage along with the host DNA gets replicated. There is this significant characteristic of the bacteriophage in the host because it has been integrated inside the genetic material. This is the first half of your lysogenic cycle, where there is an attachment of your phage to the bacteria. The first step is called as attachment. The second step is called as entry. Just like the lytic cycle, there is entry. Then in the third stage, there is something called as integration, which is not there in the lytic cycle. Integration happens in lysogenic pathway only. Here, there is an integration of your phage DNA inside the genome of your bacteria, and there is a recombination that is being created. Now, the progeny that is being synthesized by this particular host will carry the genetic material of the bacteriophage as well. Then how is the pathogenesis formed? As I told you, this is just a part of the lysogenic pathway. When there is a particular time is being produced, all the environmental conditions, they will switch to lytic cycle. Here, as I told you, there is a phage material that is being integrated inside the host DNA. Then that continues replicating, where the progeny will contain the viral DNA as well. But once the appropriate environmental condition appears, the prophage detaches from the integrated chromosome. It detaches from the host chromosome and switch it to lytic pathway. Here, there is a switching or removal of the phage DNA from the genetic material of the host, and thus switches to lytic pathway, where there is synthesis of newly synthesized proteins and subunits of the viral. There is assembly and there is maturation and lysis. So lysogenic pathway contains integration of your prophage into the host chromosome, and as that host chromosome divides along with its genome, even the prophage also divides. On set of favorable condition, the prophage detaches from the chromosome of the host, switches to lytic pathway. This is what I was trying to explain you, where there is an integration of your viral genome inside the bacterial chromosome. When the prophage exits from the bacterial chromosome, it switches to lytic pathway. So this is a comprehensive table where the bacteria shows lytic and lysogenic pathway, where it starts from here. The phage material attaches to the bacterial cell, the nucleic acid enters inside the bacterial cell, and there is no integration of the bacteriophageal DNA So it is lytic pathway. But when it's integrated inside the bacterial chromosome, it is lysogenic pathway. Then as the bacteria divides, even the prophage also divides. But on set of favorable condition, your prophage DNA detaches from the bacterial chromosome, switches to lytic pathway. And that is all we have in today's class. Thank you.